right? This is going to be our donor cell. This is our recipient cell. This is meant to be that double-stranded, single circular chromosome of the donor, shown in red and orange. And then this is the recipient DNA. And what we're looking for at the end of this process is for this recipient cell to have some of that DNA and to become recombined. So we'll watch that occur, right? So this is the most simple example of that, where a cell degrades, right? It dies. You sprayed calcite on the countertop. Because the DNA is not protected by the cell, it fragments, and cells actually have these proteins on their surface whose job is to bind DNA from the environment. Bacteria actually do that. They grab DNA, and these little REC-A proteins cut and add that piece of DNA to that cell. Now the question is, are you going to keep that piece of DNA, or are you going to cut it out and replace it to complement what was already there? That's one option. That's not a recombination because that cell is the same. But this time, you get a recombination event, and the recombinant recipient cell now has a new phenotype. Now, of course, new doesn't mean better. You could have had something better, and now you have this in its place, which maybe isn't a cool thing, right? But for bacteria, this is a gamble worth taking because this is just one of the billions of cells that exist in solution, right? And if he dies, that doesn't mean all of the bacteria die. That means he doesn't pass his genes on, the other guys do. But if that was a benefit to him, because let's say it was an antibiotic-resistant gene, and he's living in a person who's taking an antibiotic, now everybody else dies, and he knows how to fight the antibiotic. So that's how that becomes functional, right? It has to become part of the chromosome in order for that fragment to become functional. And the way that it does that is through those REC-A proteins, right? So the REC-A proteins actually sit on the surface of the cell, grab DNA, bring that DNA into the cell and utilize it, right? Put it inside the chromosome. And once it's in the chromosome, we have promoters, so we can get transcription of the genes. We have an origin of replication so that we can copy the gene, and the gene becomes functional. So one way that DNA can get into a chromosome and become functional is using REC-A proteins, right? The second way is a little more exciting, and that is through the use of what were coined jumping genes. And they were actually discovered by a plant biologist named Barbara McClintock, and nobody knew how smart she was because at the time that she made this discovery, everybody laughed at her and told her she was ridiculous, and then she got the Nobel Prize and people were like, just kidding, Barbs. So she hypothesized, she looked at corn, right? She looked at single kernels of corn on an ear of corn, and she noticed that in those kernels of corn, there was like a speckly color. Part of the kernel was orange, part of the kernel was yellow, part of the kernel was white, and she thought to herself, they should all be the same color because, each kernel of corn is one seed, and it's the result of one single pollen strand. The hairs on corn are actually tubes that pollen can go in, and they go, and there's one for every seed there. So she thought, that shouldn't be different. Everything inside that kernel should be the same because they all came from the same cell. And so she said, you know what I think's happening? And how she thought of this, I have no idea. She goes, I think a piece of DNA is jumping inside the pigment genes and disrupting them. Right? And that's why she got the Nobel Prize, because she's smarter than any of us, because we wouldn't have thought of that, right? But everybody was like, come on, Dabs, that doesn't even make any sense, right? Negative. And it turned out she was right. There are these things, right? Their cute name is jumping genes. Their official name is transposon. And a transposon is actually a fragment of DNA that will jump in and out of chromosomal DNA. So again, this is a process that's nice to see in motion. So this is a picture that you have in your notes if you are in the habit of printing them out. This is a video that you can access, again, if you would like, by, I'm going to make sure I turn up the volume right now. You can access this video by going to the Chapter 8 notes and going to the animations at the end of them. And this, notice is a jumping gene that carries with it resistance to the antibiotic kanamycin, and this is an actual example of a jumping gene.
Complex transposons consist of two simple transposons with another sequence of DNA between them.
Aren't they cute?
This other DNA sequence is often a gene that gives some selective advantage to the organism containing the transposon, such as antibiotic resistance. For example, the transposon shown here is TN5, which consists of two IS1 insertion sequences and a gene for resistance to the antibiotic kanamycin. The ends of a complex transposon are insertion sequences, for example, IS1. The insertion sequences are capable of moving or copying themselves independent of the rest of the complex transposon. But insertion sequences that carry along an advantageous gene, such as the one for kanamycin resistance shown here, are more likely to survive to be copied when the bacteria reproduce. The element in the middle of the complex transposon cannot move on its own, but it usually confers some advantage to the transposon if it's brought along as the transposons jump or are copied from one place to another. Therefore, insertion sequences flanking such useful elements are more likely to be copied when their host organism reproduces.
All right, so that's another way that DNA can become functional, right? It can actually insert itself into the chromosome, and it makes it a very dangerous thing from our perspective as the host of these bacteria, that they can jump out and go to different cells. And so we know about this because this has been seen in hospitals, right? Bacteria readily share kanamycin resistance by the use of this transposon jumping in and out of bacteria. Yes?
So is this like how they say some like diseases or viruses have become susceptible to what are those antibiotics because of these jumping genes?
Yeah, some of them. But we want to make, so susceptible actually means that you can use the antibiotic. Resistant.
Oh, I'm sorry.
Yeah, no,
resistant.
I figured that's what
Yeah.
you meant, but just in case someone else hadn't heard that word, I didn't
Yeah.
want something else. So susceptible is sensitive. Those are both used interchangeably, but yes, this is a mechanism of antibiotic resistance. And if you think about it, in a hospital, what are you constantly doing to the surfaces that you're working on? You're spraying them with disinfectant, which is lysing open dead cells and freeing that DNA up. And if they've been in the hospital, they're more likely to have kanamycin resistance anyway, right? Because survival of the fittest says, if you're the bacterium who has resistance, you're gonna survive better in a hospital. And then a new person comes in and they cough out live bacteria and they land on the countertop and they go, hey, look at that, that's some kanamycin resistance, I would like to have that, right? And they go, whoop, and they put it in their pocket and they become resistant too. So yeah, and we're gonna talk a lot about antibiotic resistance in, I always forget, I think it's chapter 20. 15 we talk about pathogenicity, 20 we talk about antibiotic resistance. So these are two ways that DNA becomes functional when it gets into a cell. So now that we have that in our tool belt, we can start talking about those three mechanisms for getting the DNA to the cell, right? So the first and easiest is transformation, and you're actually, right, which is nice, because you're going to be doing a transformation. We're going to be transforming bacteria, not with antibiotic resistance, because that would not be ethical. We're actually going to teach them how to glow, which is kind of fun. They extracted a gene from deep sea jellyfish that fluoresces, and we're going to insert that gene into bacteria and see if we can make them fluorescent. So we're going to see that phenotypic change. And it's a pretty easy procedure-ish. Like you're going to have to do, it's going to take you maybe like 10 minutes. Oh my goodness, right? But it's a pretty easy procedure because bacteria, right, they have those proteins on their surface that want to bind DNA from the environment. So that fragment of DNA floating around in solution, right, lands on that piece of DNA because everybody's moving around in solution. The cell says, hey, that looks like that might be fun, grabs it, uses that REC-A protein, right, or if this is a transposon fragment, comes into the cell, transposon, boop, right in there, otherwise REC-A proteins, cut it in, and it changes the phenotype of the recipient cell. So we don't have little a's and big A's together, right, because bacteria are always haploid, but we still will use that big A for one form of the allele, little a for other form of the allele. We won't call them dominant and recessive because we only have one, so there's no possibility they're going to mask one another, right? So the recipient cell was big A, and it received a fragment of DNA that contained little a, and little a got incorporated into it, and so now it has a different phenotype. And that phenotype could be a benefit to the cell. That new phenotype could be a detriment. You could have been better off before, right? It's the environment that decides. Even, it's kind of strange to think about, even receiving antibiotic resistance is only a benefit if you're living in an environment where there are antibiotics, right? Because ATP is precious, right? We talked about how much it takes to make ATP, how expensive every cellular process is, right? At least 3 ATP for every amino acid. If you're gonna make an enzyme to kill an antibiotic, and there's no antibiotic there, you're wasting your time. You're wasting your ATP. So you're not the best fit in the environment, which is actually super good news, because how do we reverse antibiotic resistance? We just stop taking antibiotics, right? We monitor the way that we take antibiotics so that we're not creating a situation where that's the best fit bacterium, right? So this could be good, this could be bad. We don't know. What we're talking about in chapter 8 is just, this is a change. It's the environment that decides whether or not the change is beneficial, right? So let's say that this was antibiotic resistance, right? Let's say he used to be susceptible and now he's resistant and he's cruising around with everybody else in your throat. And he has no selective advantage because you're not on antibiotics, so he's alive, his buddies are alive, everybody's cruising around, and then you get an infection and you start taking antibiotics. See, the environment changes, and now he's better suited. But it's an environmental thing, right? And it's important to realize that bugs, it's, you know, they don't have an intellectual capability. It's not like they see the antibiotic coming and they go, I need to figure out a way to get rid of that thing. They just already have a way to get rid of it. And you've introduced it and they go, oh, I know what to do with that. And they kill it, right? They don't create antibiotic resistance as a response to your using an antibiotic. They are resistant already. And you might wonder why, and this is kind of a spoiler alert, but the reason they're already resistant is because antibiotics are produced by microorganisms. It's a way that they compete with one another for space, right? They don't want to share, so they actually produce antibiotics and they kill off the competition. So because they've been using them naturally for probably as long as they've been around, there are bacteria, right, that are becoming resistant to them. It's just that when we use them clinically, they become a bigger issue for us, because we don't care what's going on in soil microorganisms, right? We care about the microorganisms that are living in our bodies. So, notice transformation, easy peasy, right? DNA gets bound to the cell because it's just floating around in solution, and either RECA or transposons get it into the cell. Now we are going to hedge our bets even further with our transformation and really make it likely by not even having to worry about getting DNA into the chromosome, because we are going to give them this green fluorescent protein gene on a plasmid, right? They don't have to do anything. It's already got its own promoter. It's actually going to be in an operon, so we can even turn on the production of the green fluorescent protein or shut off the production of the green fluorescent protein, because it's an inducible operon. It will replicate the plasmid itself. It will turn on the genes itself. All it needs to do is get into the cell so that the cell can provide the pieces that it needs to make that, right? So in this example here, we have a plasmid with a new gene, right? So rather than being an alternate form of a gene that bacterium already has, this is a new one, right? So I give it the name E. So gene E comes in, and gene E, this is the end of the story. He does not have to get into the chromosome. He can stay here in the plasmid because the plasmid is self-replicating and it has its own promoters. So it will replicate every time the bacterial cell undergoes binary fission, and it will transcribe and translate the genes that are on it, and so you will see that phenotypic change, right? So for us, we're gonna use E. coli, and we're gonna transform them, and for us, E will be a green fluorescent protein gene. And we're gonna see if we can make them fluorescent. Yay, right? Just in time for Halloween, so that'll be fun. But you don't get to take them home. All right, so that's transformation, right? So notice in both of these cases, the donor cell is nowhere near, right? Because this is naked DNA or free DNA in solution, and that's what you want to be thinking about, right? Naked DNA in solution getting into a cell. We skipped conjugation, by the way, because it's the most complex, so we're going to save that for last. So we're going to skip to the bottom of the list and go to transduction. Now, transduction is virally mediated, right? And for the purposes of this lecture exam, what I want you to know is that it's virally mediated. When we study viruses in our next unit, it's going to make more sense to you how this can be. But because we're going to talk about what viral infection looks like. So for right now, you just need to think, okay, that's a virus, does that, right? So here's a virus. Remember we talked in the Hershey-Chase experiments about particular kinds of viruses called bacteriophages. They're bacteria eaters. Bacteriophages replicate like all viruses by infecting a cell and making the cell make their babies for them, right? So they will either go into the cell completely, or they will just release their DNA, or it turns out RNA, their genetic material, into the cell, and then the cell will make babies for them, right? So that was where Hershey and Chase were trying to figure out, right? Which part went in? Was it the protein coat, or was it the DNA inside, right? And they found that when they had the radioactively labeled DNA, that's what ended up inside the cell. So things get really crazy inside of a cell when viruses are replicating, right? It's a madhouse. You have like millions and millions of viruses are actually inside the cell, right? So this picture shows, 1, 2, 3, 4, 5, 6, 7 viruses being replicated. That's not actually what happens. The virus comes in, and to make sure that he has the attention of the cell that he's infected, he takes the cell's chromosome and chops it up into bits. And that way he can make sure that the cell is not going to do any of the cell's business. Its job will be to make baby viruses. So notice kind of step 1 is get in there and chop up the DNA and make it totally useless. So now the cell can't do anything on its own. All of its recipes have been shredded. Right? It's like taking its chromosome and running it through a paper shredder. No more, bacterial stuff going on. Now, the bacterium will just make baby viruses, right? So it'll make a bunch of baby virus DNA. It's sort of like an assembly line, right? It makes all the DNA, and then it makes all the parts, and then it stuffs the DNA into each of the parts. But remember, it's not 7 viruses, it's bazillions, and it's super crowded. And so sometimes, when they're making the baby viruses, they'll accidentally grab a piece of that shredded bacterial DNA and shove that into a baby virus. So here we have the donor cell. He has purple DNA. The virus has kind of a pink DNA. Notice, right, 2 of these bacteriophages don't have viral DNA in them. They have bacterial DNA in them. Which doesn't matter to the outside, it can't tell, right? All it knows is my job is to go find a cell and inject my DNA into that cell. And I don't care what it is because I'm just an automaton, right? My job, now that I've got some DNA inside me, is to roll over there, punch a hole in the cell wall of that bacterium, and kind of vomit my DNA into them. They're kind of like little baby syringes, actually, these bacteriophages. If you look at them, they actually inject their DNA through. They have these weird kind of sharp projections that stick to the cell wall and then punch a hole through it so that the DNA can go inside. So you can imagine for the bacterium that gets that guy attached to him, he's going to make a bunch of baby viruses. He's going to make a bunch of baby viruses. He's going to make a bunch of baby viruses. He is and he is. But these 2 are going to inject bacterial DNA into a cell. And that cell is not going to make baby viruses because what directs the replication of the virus? We know from Hershey and Chase it's not the protein, it's the DNA. So if there's no viral DNA, there's no viruses, right? So what comes over is some other kind of bacterial DNA. So it gets into the cell, it's a fragment of DNA, right? So there are two options, Rep-A or transposons, to get that DNA into the chromosome. Because it's a fragment, it can't operate on its own. It has to get into the chromosome. And if it gets into the chromosome, you end up with a recombinant cell, who is recombinant because a virus brought the DNA to him, right? So transformation, naked or free DNA just picked up by the cell. Here, you actually have a virus punching a hole through the recipient cell and injecting DNA into the cell. And again, right, we don't know whether that's a benefit. It's just a change. The environment decides whether it's a benefit. Could be a bad thing, right? Could be that you get, sucker punched, and now also you have some piece of DNA that's worthless to you and actually makes you worse off than you were in the past. But we want to think, right? Our question here is, how does the DNA get into the cell because a virus brought it? Yes?
So the resulting cells, are those infected cells?
No,
Because
they're
of the
not
virus?
infected because it's not viral DNA.
Oh, it's bacterial.
Yeah, so if you print this in black and white, make sure that you make some kind of note, right? That it's not the phage DNA, it's the one with bacterial DNA that is infecting this cell. So it is infecting in that the protein comes over and injects the DNA into the cell. But if it's infecting with bacterial DNA, not viral DNA. So this is not going to cause infection of that bacterium. It's going to give it a new bacterial trait, whatever that trait is that it brought over from the bacterium that it killed.
So where would the viral DNA go?
So they're just going off and infecting other cells.
Oh, okay.
Yeah, it's basically a mistake on the part of the virus, right? He doesn't want to do that because now he's wasted all his time making a viral capsid that he put bacterial DNA in. But it's just because it's so crowded that they made a mistake.
Okay, thank you.
Okay, third one, most complicated, so that's the one we're going to talk about last, and that is conjugation. So conjugation, cell-to-cell contact through that extracellular structure that we talked about in chapter 4, the pilus, right? The pilus is a little tunnel through which bacteria can share DNA. And the easiest way for that to happen, simple conjugation, happens when a bacterial cell that has an F factor gives a copy of that F factor to its neighbor, right? So you got these two cells and they're cruising around, swimming, right? Playing, minding their own business, like two kids in a swimming pool. And they get close enough and this F plus cell, which we call F plus because he has an F factor. Oh, I don't like it when I accidentally pick up other people's things that has an F factor, right? An F factor, you'll remember, is a plasmid that has an origin of transfer, an origin of replication, and it has the genes to make a pilus, right, and move this whole thing into a recipient cell, right? So this means that it's self-replicating, means all it has to do is get in a cell and it functions, right? No problem. So when this cell, who is F plus, encounters an F minus cell, he's not a very nice guy. He actually grabs onto the F minus cell, punches a hole in his cell wall, and starts jamming DNA into him. That's basically what happens, right? He moves that piece of DNA, he replicates his F factor because for some unknown reason, he doesn't want to give up the copy he has. He wants to make a copy and give it to this guy. So when he does that, the recipient becomes F plus, right? Now this is a great illustration of how getting new DNA may not necessarily be a good thing for you, right? So we've talked about how much energy it costs to make nucleotides, to make peptidoglycan, to do all of the cellular work, right? So imagine, right, you're this F minus guy, you're just cruising around, somebody punches you in the side of the head and starts throwing information into you. You're like, thanks a lot, right? But the real kicker is, next time you run into a cell, you're an F plus cell, so what are you gonna do? punch somebody in the head, spend a bunch of energy making a copy of this worthless piece of DNA and giving it to them. So it's like a bad mood. You know how whenever someone's in a bad mood, the bad moods get shared, right? I mean, in my house they do. We actually have a book about it when they were little. It's called The Bad Mood. It's about a little badger who gets in a bad mood and he spreads it all over the forest, right? So it's kind of like a bad mood, right? So we got one cell in a bad mood, punches him, gives him this piece of DNA, and then this guy, now he's in a bad mood too. Now we got two cells in a bad mood, and both of them are in a bad mood because they're going to spend a bunch of ATP doing the same thing to more cells. It's completely worthless. It doesn't do anything for them. So why does it exist? Because sometimes a little something different happens. So this is very simple, right? F plus encounters F minus, which means that he has no F factor. And the end result of that friendship is 2 F plus cells. There is, by the way, a summary table in the lecture notes that you can see that statement written out in. So sometimes an F plus cell will, the plasma, the F factor will get up close to the chromosome and actually cut the chromosome and open it up and become part of the chromosome. So notice here, right, the F factor is kind of in this pink color. The chromosome is in this light purple. Notice they connect and it becomes part of the chromosome. Now, that doesn't mean really anything different for this cell phenotypically, right? When he meets another cell, he is going to punch him in the head and start sending DNA into that cell. But it does mean something different for the guy who gets punched in the head, because he's not going to send the F factor, he's going to send some of his DNA through that pilus. And so this cell is not called an F plus. Notice that doesn't say F plus. That cell is called an HFR cell, which stands for high frequency of recombination. And I don't know exactly why they gave it that name. I suspect that they were watching cells interact and do this thing, and they understood there was an F factor, the genes were there, and in this situation, there was never any recombination, but for some reason, a subset of those F plus cells, phenotypically, were giving DNA to a recipient cell that made them recombinant. So they said, well, these, whatever's different about these gives them a high frequency of recombination. The F plus have a 0 frequency of recombination, but these guys have a lot of recombination, right? And as they looked further, they realized, oh, the difference between an F plus cell and an HFR cell is whether the plasmid is all by itself as a single separate piece of DNA, or whether the plasmid has been incorporated into the chromosome, right? So those are HFR. So now we have 3 kinds of cells. We have F plus, which have an F factor, F minus, which don't, and HFR, which have an F factor that's been integrated into their chromosome. So why is it that when the F factor gets integrated into the chromosome, you end up with a high frequency of recombination? The answer to that is right here. The origin of transfer is the place within the F factor where it starts moving, right? Where replication of that piece of DNA starts and the replicated piece starts moving. But it's not the origin of insertion. It's not the same place that the DNA goes into the cell. The DNA goes into the cell over here. Boy, I'm striking out all over the place. Right? Right here. That's the spot where it goes into chromosomes and makes that HFR cell, right? So if we look at this picture, that is the site of insertion. That's the site of transfer. So it inserts here, right? Site of transfer is right here. So he's cruising along and he encounters another cell and he punches him in the head and he starts sending the DNA right here. And he sends a little bit of the F factor, but then he starts sending his own chromosome. And he would have to get through 3 million nucleotides before he would send the rest of the F factor over. And the likelihood of that is infinitesimal, right? Because we have 2 cells floating in solution, suspended by this pilus, which is long and skinny and fragile. Have you ever seen pictures of like a bridge when there's an earthquake? And this is why you learn calculus, right? So you can figure out the sine wave of the bridge as it kablamies, right? Because as things move, that becomes very tenuous and it breaks. So as he's moving the DNA over, they're both moving and the pilus breaks and they stop receiving the DNA. So they never get the whole F factor. So this is the only place where it could possibly be a benefit to interact with an F factor. It's not a benefit to hang out with this guy, because all he does is make you just like him. Now you both will spend a ton of ATP and a ton of time making an F factor and giving it to other cells that don't want it. But if an HFR cell encounters an F- cell, he will give him some of his DNA. Now, does he become an F+ cell? What do you have to be an F+ cell? You gotta have an F factor, right? Does he have an F factor? No, so I know it's little, but that's an F- cell. He's still F-. So he doesn't get any of the baggage. He's not gonna make an F, he's not gonna make a pilus, he's not gonna make DNA and give it to somebody else, but what he does get out of this bargain is a little piece of DNA from that donor cell. Now again, we don't know what that piece of DNA is. We don't know that it's a benefit, right? All we know is it's different DNA, it's different bacterial DNA. So when F+ and F- get together, the only thing that changes is that they're now both F+. The only time you have any possibility of anything different, right, like a leucine gene or a histidine gene or something like that, is if you have an HFR cell. Yeah?
So HFR cells do not have an F factor?
The HFR does.
They have a little piece of it?
They have the whole thing. The recombinant recipient is not an HFR cell. So here's your HFR. That's that guy. So he's the donor. But what he donates is not the F factor, it's a piece of his own DNA. So, I mean, really, if this was a more accurate picture, there'd be a little tiny piece of pink there, but not enough that this guy will make a sex pilus, right? So not enough to actually be the F factor. So this is a recombinant cell who has a new trait, right? And so new traits would be maybe the ability to make an amino acid, right? Maybe it's part of the tryptophan operon, right? So now he can make tryptophan if he wants to. So this is a new trait, but when F+ and F- get together, the only thing that happens is that they both become F+, right? So this picture here, right, these are both F+, assumably because he made a pilus and sent over an F factor to him. And that is it for horizontal gene transfer, right? So we have donors who donate DNA to recipients who become recombinant. And recombinant is going to be a word we hear a lot. We're going to talk about recombinant cells and recombinant DNA, right? All sorts of things, but recombinant means you have your original stuff and you have some new stuff. So in like the common vernacular, when you hear genetically modified, that's what it means. Your original stuff and some new stuff that you've been given. So as you're thinking about these, right, remember what I told you. So conjugation, right, that's cell-to-cell contact, movement through a sex pilus. When we talk about transformation, that's free DNA in solution. When we talk about transduction, that is virally mediated, and there is a summary table in your lecture notes that you can look at, kind of get that straight, right? All right. Well, I guess one thing I do want to say, and I've said this a bunch, right? But this is, when you hear repetition, you know, ooh, we actually pay attention to that, right? It doesn't mean anything is good. It just means something is different, right? It's the environment who decides what's good. And that changes. Every time the environment changes, what's good changes, right? Like when it's cold out, a sweater is good. When it's 100 degrees, a sweater is not good anymore. It's not anything to do with the sweater, it's the environment. And jeans are the same way. Jeans are not good or bad. It's whether or not they make you more likely to survive. And sometimes different things make you more likely to survive. So that's it for chapter 8. Yay, we're done with the first unit. Your test is coming. Study for your test. Don't make the mistake of being like, oh, it'll probably be super easy. And if it's not, I'll just make up for it later. Hit it hard. And if you studied so much and you're like, that was so easy, it took me 5 minutes, then you can study less next time. But it's 60 points, 70 points, something like that, 70? That's 17% of your course grade. Don't leave it forever, work on it. I'm not sure, I think, Maria, your hand was up first, but I'll get you too.
It's just multiple choice.
It is, yeah, so it's like 50 questions, it's multiple choice, you could write Scantron, and the multiple choice, I know people don't like multiple choice tests, but you have to learn how to take multiple choice tests. It's an important skill, right? Also, it allows me to do statistical analyses on the test and figure out, are the questions working? Are they asking questions that people are capable of answering, right? And it allows me to make sure that my test is fair. Because I have this huge bank of test questions, you know, I've been doing this for over 20 years, and I pull questions that at least, you know, this many percentage of people can answer correctly, right? So I know people don't like multiple choice. I also don't like listening to people babble on and have no idea what they're saying for like 13 paragraphs. I'm like, and you're like, but I wrote a lot. And I'm like, yes, and my life has been shortened by reading it. So we're not gonna do that to one another. So yeah, multiple choice.
That's the lecture exam.
That's the lecture exam. Lab exam, which is coming first, is short answer. It's not essay. It might be one word answers sometimes, right? Maybe you'll have to estimate the size of a microorganism looking at a picture of it, right? You should know how to gram stain. You should know how to streak plate, meaning like know how to do that without notes because you don't get notes, and recognize what a spore looks like and what an acid-fast bacterium looks like, right? So it could just be, you know, where's the spore, and then you draw an arrow to it, or what's this piece of the microscope or something
So
like
it's
that.
not like actual like trying to do the lab.
Oh yeah, yeah. If it says know how to do this, then you better know how to do it.
No, like we're gonna actually do it.
Yeah.
On the exam.
Mhm.
Okay.
It's a practical and a written exam.
Oh, okay.
So lab exams always have a practical portion, and that's the thing, right? If you miss the exam, you never get to take the practical, because we can't like meet up outside of class time, like in the cafeteria, and do a gram stain, right? So if you were not to be here on Tuesday, you would never have the opportunity for those points. So you want to make sure you're here for lab exams. Not that I'm looking at, because I'm assuming you're not going to be here, but be here on
Yes.
Tuesday and be here on the following Tuesday. And Shadi, what was your question?
She asked the same one.
Cool, yeah, so when you look on the study guide, it says, know how to do this, that's because you should know how to do that, right? And if it's any staining technique, right, it's the decolorizing step that you need to think about. Yes?
How many questions are on the lab exam?
I don't know, 20 something, 20 or so, something like that. And it's meant to be a 2-hour test, so you'll get 2 hours to take it, but that's giving you ample time for practical stuff. What's that?
Do we have a lecture or a lab that day, or is it just the test?
Yes, we have a lecture. So we have a lecture before the lab exam,
Okay.
and we have a lab after the lecture exam.
Okay.
The reason your lecture exams are where they are is because the lab exams are set for me. I don't decide when you take a lab exam. There's a big meeting with 8 teachers fighting over when the exam should happen, and then everybody's exams happen on the same day, so our lab tech doesn't go insane. Okay. Okay. Yeah. So yes, there will be a lecture, and then I'll give you. I usually give it on an exam day, like a lab exam day. I'll give you like a half hour between lecture and lab, so you can have some time to go back over your notes and look things over. It's not like 5 minutes after we're done with lecture, you gotta go into lab exam. But we do have both, because they're kind of treated like 2 separate courses. Okay, so today we're doing this. Yay, 4 times. Which should be a piece of cake, because you know how to use a spectrophotometer, you know how to use a micropipetter, you know how to read serial dilutions, you know how to do serial dilutions. So this is the picture from page 66 in your lab book, right? And that is what you will do every 30 minutes for a total of 2 hours. You can ponder that for 1 second while I keep the cultures and put them back in the shaken incubator so that we can get going toward our 30 minutes.
There's 25 questions.
So is she gonna watch us do it, or is she just gonna? I don't know. Like, is she gonna watch us do it, or is she gonna be like, alright, let's see your final results and see if we can do it?
What does cold temperature do? Slows them down, right? So we stop them from undergoing binary fission. So that's why they've been in the cold. So they went in the cold as soon as they were red. And now we're gonna start a 30-minute series. And then you will read the next 30-minute reading. At that point, you will take your flask out. You will try to keep it outside of the incubator for as little time as possible because 37 degrees is their optimum. And when we bring them out onto the countertop, we will slow them from that, right? Because as we reduce the temperature, they're gonna grow more slowly. So we want to have them back in the incubator as soon as possible. So when they come out, they're only gonna come out when you are ready and someone is waiting with a cuvette and a micropipetter. And you're gonna wanna help, right? This is a one-man job. A pipette man, right, is a pipette man, not men. It's meant for one person. So I know you like each other, you wanna hold tubes open for one another, negatory, right? We don't hold things open for one another. You set it on the counter, that person opens it, draws out 1 mil, closes it, puts that mil in the cuvette, draws out another mil, closes it, puts that second mil in, and then hands it to another person who is waiting with the P200 set to 100 to draw out 100 microliters and put it into 9.9 mils of water. Then you will walk back to the incubator, put your flask back in there, and now it's shaking. And for 30 minutes, all you have to do is read the cuvette, serially dilute this, and spread plate it, which looks like this, right? So that's your culture, it's already back there. This one's been diluted 10 to the -2. So now you take that same P1000 set to 100, and you go in and out and in and out and in and out and in and out of this tube to stir it all up. And then you take 1 mil and you put it into that one. And then you get rid of that tip. And then you get a new tip and you go in and out and in and out and in and out and in and out. And then transfer mil. And then you say, person number 4, here's your dilutions, plate them. And person number 4 of your team, or person number 1 again of the 3-man team, takes these dilutions and plates 0.1 mil, 100 microliters, onto each of those 4 plates, right? We're gonna use a new technique called spread plating, so I'll show you how to do that. When you're ready to do that, we'll do that together. I can show you how to do that. And then, right, we only plated 100 microliters, not to be a jerk and increase dilutions and make math hard, but so that the liquid will absorb super quickly and they'll be ready to go into the incubator. So what I'm gonna do is, while you're getting set up, I'm gonna go into the other lab and steal their pipette men so that each team has 2 P1000s and 2 P200s so that you have more to work with, right? Because we're gonna need those today. I think I'm gonna do that, I don't know. I'm for sure gonna do that in a few labs. But that's what you're gonna do. And the first time you do it, it's gonna be chaotic, it's gonna be crazy, and you're gonna be like, oh yeah, right? But now you've done it once, it's gonna be super easy the next 3 times. So the first time probably will be hectic. And it may take you the whole 30 minutes to actually get through this procedure, and then you'll be like, oh my gosh, we have 5 minutes until the next round, right? But that's okay, because the next time you know how to do it, and it's much easier. One word to the wise, and that is, when you are done with this, you no longer need the cuvette, you no longer need any of these test tubes. Get rid of them before you bring your next set. So right now, you will need 5 tubes and 4 plates Total, you'll need way more than that, but don't get all of it right now. Get enough for set 1, right, which is 4 plates, 5 tubes, one of which has 9.9 mils of water, 4 of which have 9.0. It looks like this is the 9.9 with the clear lids. And you don't have to put inoculation labels on these because we're not doing anything with them. They're going in the trash. So if you promise to clean your marks off, you can write -2, -3, -4, -5, -6 with one of these China pencils, and before you put it in the morgue today, just rub on it with a piece of Kim Wipe and it will wipe that mark off. If you don't do that, then you're going to be very sad because we're going to have a chat about how you're making more work for other people because you're too lazy to do your own stuff, right? So, when we discard today, doing something a little bit differently, so just make sure that you follow these protocols.